All right, thank you. Thank you all for coming. I'm delighted to be here. I am just like to thank Stuart and Becky for sponsoring this. And Stuart came to myself and Wolfgang and Samik Roy-Chaudhry. The three of us kind of put this together, so hopefully this will make sense. For those of you who may not know me, I'm a cardiologist, a heart failure, heart transplant cardiologist. I came to OSU in 2012 to direct the Division of Human Genetics. It's a bit of a different move for a clinical cardiologist, but so far it's going well and I enjoy it. I have no conflicts. I will provide a very broad overview for hopefully a diverse group here. You all know that human genetics and genomics is, I envision it always as a conversation between phenotype and genotype. That is the essence of what we do. Of course, phenotyping what we observe, clinical studies of diverse kind, depending upon what your phenotype of interest is. And of course, genotype, DNA sequence, we focus on sequence. There are a lot of permutations to sequence analysis, but that is molecular studies. It is this arrow between, this communication between these. I'm more clinical, so phenotyping is critical, and of course, as you well know, you can have the most fabulous sequence analysis, but if your phenotype doesn't make sense, you got nowhere, and just the opposite. You can have fabulous phenotyping, but if you don't really have state-of-the-art genotyping and genomic science, you can't make headway. So it's a critical, fundamental concept. So this precision medicine term, some people love it, some people aren't in love with it as much. What came before that term? I would say just medical genetics or genetic medicine, which is, as you know, the specialty of medicine that involves the diagnosis and treatment of hereditary disorders. This is a picture of a classic genetic syndrome, this incredible arm span of someone with Marfan syndrome. This is classic genetics, and precision medicine certainly includes medical genetics, but more recently, really in the last 10 to 15 years, this term of genomic medicine, which was this concept of an emerging practice, using genomic medicine. The problem was we didn't really have the data we needed. GWAS came along, it made some incredible impact, and of course led to the 23andMe types of approaches using GWAS data, but again, insufficient. Personalized medicine, a more recent term. To me, as a physician, everything is personalized, but what is now, of course, the term du jour is precision medicine. From a genetics and genomics perspective, to me, it is most simply genetics and genomics information integrated more broadly and more precisely, more comprehensively, really from the get-go, into clinical medicine. The central concept is the same for all of genetics and genomics. It's identifying and managing genomic cause or genomic risk. Those words, cause versus risk, of course, if you talk to geneticists, have fairly different meanings, and of course, when you get into statistical analysis, they also have different paradigms. I will not discuss that, but those are the central concepts always. And of course, we try to make this specific for an individual or family as much as possible. But really taking a step back, so what's really that new about it? I do think it is, and I'm talking now from a clinical perspective, the clinicians that I work with, it really is a paradigm shift in how we view not just disease, but also health. And of course, not only in terms of just defining something clinically. Again, in the cardiovascular universe, you grab an echo or you grab a coronary angiogram. Instead of just defining it clinically, you begin to imagine immediately from the get-go putting in genomic information. And we really are literally rewriting the textbooks of medicine. I was asked to co-author a chapter in the Brownwell textbook of cardiovascular medicine a couple years ago. The reason they asked me was there used to be the genetics chapter up front or a couple of genetics chapters and then the clinical cardiology and the cardiomyopathies, and they wanted a chapter where it was all put together. And that's what I did for them. We are literally rewriting the textbooks, not only of cardiovascular medicine, but of cancer and everything else. Now, underlying all of this, everyone in this room has probably seen this slide. If not, it's fundamental background critical information. Notice the vertical axis is a log scale, And basically, I hope this deflection point is real, but it suggests that the human genome sequence cost is closing in at $1,000, and that's pretty close depending on what your coverage is, et cetera. It is this technological transformation, and it really is transformative, that has permitted all of this to move ahead. Conceptually, we were there a long time ago, but we simply couldn't get it done. We can get it done now. So we use information from DNA sequencing, again, to identify risk or specific cause, to guide prevention and early intervention, and enable specific therapy. I'm not going to discuss gene therapy. It's a very exciting concept. It's alive and well in Columbus. And it is applicable, in my view. There are really three domains: heritable or germline genetic disease, cancer, somatic genetic disease, and pharmacogenomics. Tailoring drug choice, dose, of course, to the individual variation, critically important. That is the theme. These three themes then become the components of the discussion for today. I think it also defines a movement. Of course, nothing political happens in the United States these days. Just kidding, if you pay attention to the news. This is a huge new impetus for federal funding. You may have heard of the VA Million Veteran Study. This has been around already for 2 or 3 years. The Precision Medicine Initiative, I'll speak a bit more about that. You may have heard in the last State of the Union message, just 10 days ago, the quote moonshot of Joe Biden. This is a picture I pulled down this morning off the web. This is from a Science News release yesterday evening. This was the first, the U.S. agency chiefs meeting on Monday. Here's Joe, here's the president, and these are the federal government agencies. What's going to happen with this is not completely clear. May not be new money. There might be some new money, would go to cancer. That's great. I won't say more about it, but again, just to say that this is in the news. This is a movement. But this goes well beyond the United States. The UK 100,000 Genome Project. If you haven't seen the thing that kicked this off, you need to see it. Here it is. 21st century businesses will rely on American science and technology, research and development. I want the country that eliminated polio and mapped the human genome to lead a new era of medicine, one that delivers the right treatment at the right time. In some patients with cystic fibrosis, this approach has reversed a disease once thought unstoppable. So tonight I'm launching a new precision medicine initiative to bring us closer to curing diseases like cancer and diabetes, and to give all of us access to the personalized information we need to keep ourselves and our families healthier. We can do this. I stopped the clip right there. You can see everyone is standing. When's the last time you've seen that at the State of the Union address? This is a political movement, and of course, as you well know, if you watch West Wing and all those other presidential shows, to get something in the State of the Union message really is fairly monumental. And there was, as you well probably know, if you don't, you need to know, there was probably a year of very intense effort before the State of the Union message last year to get this in, and then a lot of follow-up. So the president then, 10 days later, on the 30th of January of 2015, had a ceremony at the White House, invited key people who had been helping with this, and then here's another clip.
And that's why we're here today, because something called precision medicine, in some cases people call it personalized medicine, gives us one of the greatest opportunities for new medical breakthroughs that we have ever seen.
Whether you believe it or not, the president said it, so.
Precision medicine is now. We're already using it. It's already affecting people's lives, and we know it works.
Key point, it's already here. We are using it, and part of what we do, at least in human genetics and genomics, is we shift or move what we are already doing into the concept of precision medicine because it makes sense. This is As you, again, if you don't know, it's critically important you understand that the NIH has spent enormous time preparing for this precision medicine initiative. Francis Collins, there's been, of course, a blog and a website. You can sign up for all of this and get all this information coming into you. There was an initial down payment of like $230 million. How much more will be put to this is uncertain. This was a report, if you're not familiar with it, this is in September. It was the working group report. It's about 60 or 70 pages. Precision medicine is an approach to disease treatment and prevention that seeks to maximize effectiveness by taking into account individual variability in genes, environment, and lifestyle. It's a larger definition that I will use, at least for our work here. And a second comment, precision medicine seeks to redefine our understanding of disease onset and progression, response, and health outcomes. More precise measurement of molecular, environmental, behavioral factors that contribute to health and disease. A very big palette of everything from basic science to behavioral and everything in between. And that's what this movement is trying to take on, again, for at least for purposes in my thinking. I always think of what can be attained now, what is directly in front of us, and to some extent at the Ohio State University Medical Center, where we have phenomenal patient resources, how can we impact patient care and then integrate that into translational medicine?
Today, the Prime Minister is once again turning to our scientists to help Britain become the first country in the world to introduce high-tech DNA mapping for cancer patients within the NHS.
So it turns out that the UK 100,000 Genomes Project started a couple years before. It is an important project. It is based within the healthcare system. As you know, it's a unified single-payer system, sequencing 100,000 genomes, and they're focusing on cancer. It's also a family-based approach, which is a bit different from the U.S. approach. But just to let you know that this is not just sort of a one-off NIH thing. This really is, I think, in many respects, a global sense. Here's another clip for you.
I'm Sir John Chisholm, and I have the great privilege of being chair of both Nesta and of Genomics England. And from that came the thought that maybe this is the time to make the next big step forward in capturing genomic information and making it really relevant to routine healthcare.
I show you that clip, routine healthcare. Now, if there are any clinicians in the audience taking genomic information and putting it into routine healthcare, talk about the moonshot. If we ever get there, I will be not only amazed but impressed, and ultimately that is where this needs to go. And maybe the National Health Service in England with this 100,000 project might actually help us get there. So central concepts. Precision medicine is based on large-scale genetic and genomic sequence information. You know this. It is essential. You have to have great sequence. Another concept, the best precision medicine programs blend, even superimpose, what traditionally have been considered separate clinical and research activities. That is, moving ahead, and that's what we have been doing in the Division of Human Genetics. You'll hear a bit more about that, something called the precision, or actually we call it the Genomic Medicine Initiative. Kevin Sweet will review that for you. Basically, any clinical activity we have at OSU, if we can superimpose research objectives so that they become as much as possible one and the same, that's where we need to be. And then our research activity, when you have that type of activity, how do you define what is research versus clinical? In my view, whatever you feel you need to consent someone for to do, that becomes research. So it becomes defined by literally the documents, the protocols and the documents. And again, we've spent in the Division of Human Genetics a lot of time in the last couple of years thinking about this. There is a proposed Center for Precision Medicine. The components, heritable disease, acquired disease, cancer, predicting drug effects. The functional domains, as you might imagine, clinical, research and scientific, training, education, administration of funding, marketing, and fundraising. The key investments I think we need are human capital, clinical expertise, statistical genetics, genetic epi, data management to help us move this ahead. Whether this will happen or not, we'll see over the next few months. The components, of course, heritable disease and prevention and treatment. We identify genetic risk. We've been doing this for a long time. We do a lot more of it now because sequencing is available. We modify risk factors as possible. Example, if we find FA, familial hypercholesterolemia, an easy risk factor to modify is cigarette smoking, a horrible combination. We implement clinical surveillance to detect a disease phenotype for early intervention. Examples would be, in my work, dilated cardiomyopathy, or in cancer, Lynch syndrome. And of course, with surveillance, you detect early disease, then you intervene. Of course, you use precision therapy if it's available. President Obama mentioned cystic fibrosis, and as you may be aware, there is a specific drug for a specific genotype that truly has it's really transformed these patients who were really sick and dying into reversible disease. Acquired disease, of course, we identify disease-causing variants, looking for specific functions and pathways, and then interrogate those with specific treatments. And of course, we predict drug effects with pharmacogenomics for efficacy and risk of adverse reaction. Those are the components. Let me finish up with comments on what a genetic evaluation is for heritable disease. If you know this, great. If you don't, it's important. We do a risk assessment for a specific phenotype. We always do a comprehensive family history. We do clinical screening of relatives, always. We do counseling regarding the condition, genetic inheritance, phenotypic features, the age of onset, symptoms, outcomes, the role of genetic testing and profiling. And then, of course, if testing is indicated, we do molecular genetic testing. We do additional evaluation and therapy as necessary. Who's a genetic evaluation for and anything special about it? Again, the central purpose for heritable disease is to assess risk, identify risk. The patient in front of you, the proband, we call that person, is important to figure out what's going on, phenotype that patient, and do genetic analysis. But that patient usually, in most cases, represents the family, the at-risk relatives. So a genetic evaluation is transitioning for most clinicians who don't think of, certainly myself as a clinical cardiologist, the transition in the last 15 years was this sense of that patient in front of me, that's my training, versus that patient is simply the proband, the first part of the family, which is the part that needs evaluation. So we have to think of the patient turning that into the proband of the family at risk. It's a critical transition from one individual to understand the family and the pedigree that may have a lot of disease. There are cardiovascular disorders with genetic testing available, as shown here. As you may know, this is a rapidly emerging field. We now have panels of several dozen genes for each of these categories. There are other disorders, of course, heritable cancers, as well as heritable neurological diseases, eye, and a variety of others, cystic fibrosis we've mentioned. So let me finish up. In summary, precision medicine, in my view, is the most thoughtful and broadly based approach to define, understand, and intervene in human health and disease, leveraging all that we have known about genetics and genomics previously for both health and disease, but also coupling this with this phenomenally powerful new sequencing resource called next-generation sequencing. I'll stop and thank you again to the organizers for the opportunity to present, and I think Wolfgang is next.
Thanks. Is this on?
This one works.
Hello? Yeah.
There we go. Well, thanks. We have two presentations from our group, so I will not show slides, and I will keep it short because I would like you to ask questions also before we go into the next session. So I think there was an excellent introduction into precision medicine, the way we understand it today. And I want to start out in pointing out a leap of faith that the president made when he said, now we can cure cystic fibrosis with a targeted approach. Well, I accept that's precision medicine, but that's a monogenic disease. And by the way, a problem for society is that that treatment now costs $200,000 a year. But those are other issues we don't want to discuss today. And then he said, well, now we apply precision medicine to diabetes and cancer. So all the common diseases that are the major burden for society overall and cost the most. And the problem with that is those diseases are highly multifactorial. The population heritability for such a disease is in the order of maybe 40 to 50 to 60 to 70%. But we do not know exactly what genetic variants and how that actually comes about. And the reason for that is there's a lot of interactions. They're multi-genes, they're multifactorial, there are family reasons, and so on and so on. So we know the extent of the genetic, of the heritability, but we do not know what's causing it. And that's called the missing heritability. And so now we go into the new era of GWAS, where we measure 5 million SNPs and impute another 4 million SNPs, little variants in the genome. So surely we'll then understand how this all works. And the answer is it doesn't. It typically explains in additive models where you add each single variant that contributes a little to the risk, it accounts for maybe 10 to 20%. So that's the missing heritability. And I think we have to ask the question, how do we overcome this? What are the major obstacles that need to be overcome? And this is really the subject of research. I'm representing the Center for Pharmacogenomics, where we try to do the basic research and then see how it's been translated. So number one, among all the SNPs and the genetic variants we find out of GWAS, we know maybe 10% of them that have any impact. We know what they do or whether they're really the causal variants. We do not know the mechanisms. So that's a major problem. And Dr. Danjing Wang will talk about the drug metabolizing enzymes. We think that this has been studied for 30 years, and it's quite clear to us that we actually have not even started to resolve some of the problems in understanding the genetics of just those genes. And she will talk about the CYP3A story. So that is an impediment, because if you only have a surrogate marker and you don't know what it does and where in the body it's active and so on, it's really hard to extrapolate into understanding the whole. Number two is that over 90%, maybe over 95% of those variants are not the ones that are typical in monogenic diseases such as cystic fibrosis, where there's a single variant that changes the conformation of the protein cannot make it to the surface of the cell, and therefore you don't have the CFTR on the cell surface. And so the drug which President Obama mentioned binds to that protein, stabilizes, and allows it to come to the surface. So this is just an exception. 90% are variants that sit somewhere along the genome. They are outside the protein coding regions. Those exons account for only just a little more than 1% of the human genome, and all the rest is outside. And they can affect not only the protein coding genes, but the now 50,000 annotated genes that make other RNAs, the non-coding RNAs, including then downstream microRNAs and SNO RNAs and so on. All these are quite important players, and we have to sort this out. Of course, we have the data now to begin addressing this. So the second point is that these variants are really the ones that are, for the human race, are under evolutionary constraints. We have to respond to the environment. And how is that happening? it's easy to understand for xenobiotic enzymes or transporter that will respond to the environment. And so you can see a lot in the cytochrome P450 family that there are a lot of variants that are frequent, and they look like they're under evolutionary selection. They have a signature of that in the genome. So these variants are selected. They are normal variants because they would be otherwise not as frequent. So we postulate there are many genes that have frequent variants that are under evolutionary selection, maybe balancing selection, though it's not a sweep. You have 30, 40, 50% allele frequency. And these are the ones that then determine how we respond to drugs. Drugs is a new environmental influence, and the epigenetic factors involved. So to account for that, we cannot find these variants easily because in GWAS, all of medicine looks for risk of disease rather than the stabilization of the status of the health system, so the resilience. These are variants that are there for resilience, but they can turn against you if the environment changes. So that's point number 2. We need to find variants that are not visible in GWAS. Those are the ones that may be the most important, and they're frequent, and they have phenotypic impact, but we're looking in the wrong hole. We're looking for risk for disease. Third point is that in evolution, rapid evolution for the human race, if you look at the expansion of the number of folks on this earth, it's terrifying or wonderful, the way you look at it. So I kind of don't like masses beyond 1 million around me, but it's never selection of one gene or one variant alone. It's a selection that's based on the environment that affects the modification of the chromatin, and Dr. Wang is going to talk about that portion. And so there are dynamic interactions. That's called epistasis. The GWAS people deny the existence or the importance of epistasis in risk for disease. Why? There is a huge study on schizophrenia where they studied about 200,000 people and found 108, and now it's already up to 130 risk loci, and then compared them and see whether any pair shows any evidence of epistasis, dynamic interaction, other than just being additive. They didn't find anything. So I think they're looking again in the wrong hole, because the chances that a risk allele at relatively low frequency is going to be under epistasis, under evolutionary selection, is relatively low. So we need to rethink what we do in personalized medicine or precision medicine. If we think about cardiovascular events, for instance, a myocardial infarction, let's assume statins protect 50% of the folks. 50% are not protected. When I tell this to some folks who are not in medicine, they're totally shocked. But even in medicine, I think we're also shocked, because this is our $20 billion industry. So 50% effective. If we bring this to 60% by understanding better how to give those drugs, out of 1 million people, that will be 100,000 people. That's very, very high, but that's not precision. No. So an incremental understanding will bring us to very important advances in medicine. Now, let me close and just say that, so we have one presentation by Dr. Wang after this, and then we also will look at the use of the databases that are emerging at a rate that's absolutely incredible. So we have thousands of GWAS. So that means each GWAS is a study of anywhere from 1,000 in meta-analysis now up to 100,000. And now, as mentioned by Ray in England, they're pretty good at this actually. They want to go to 1 million, So you have 1 million people with 4 million SNPs that are measured with all kinds of phenotypes, now with a better understanding, prepared so well that it's not only a genetic study, there will be understanding of which drugs are taken, age of onset. All the clinical trials we want to do are in these databases. That's not just genetic. So because of that, our group has taken initiative in beginning to download all these large databases. And so Dr. Beth Berry is going to talk about that and what we do with these databases, because I urge everyone to consider the richness of these databases. They're not precision medicine. We have to extract information from that. That incrementally increases our understanding, but we have to apply the right concepts in analyzing these data. So she will give you some examples as to how we do that. And now, how much time do we have? Do we have some?
Perfectly on time. It's 9:40.
9:40. 10 minutes discussion. So I'm sure that there are many people here who are passionate about improving medicine, and there must be many ideas about that. So I invite questions and
Comments, yeah.
And let me just say that we are videotaping this for people to look at later, and also so people can hear you. There are microphones in front of you, and there's a white button on the right. So if you just push that right button, and then you can speak your comment, and people can hear. Just remember maybe to push the button to turn it off when you're finished. So please, comments.
Yeah, I was wondering about the the issues in the context of precision medicine in relation to treatment issues and improvements in treatment. And so when I look at historically how we've made advances, I guess initially there was predominantly a focus on environmental issues and lifestyle issues, and then that they exclusively switched to a focus on gene variability, basically ignoring what everything we'd learned before that. And so I was very interested to see that the precision medicine initiative at least clustered the three together in order to make advances. But yet when the central concept was presented, it looked to me essentially a focus again on genetics, genomics, but then in a larger scale. So, I mean, this is all new to me, but I'm just curious, how are we going to make an advance if we're going to exclusively look into genetics again?
We should not exclusively look into genetics. And I think I try to make this point that these big studies have other information. But just an example, here at OSU, we should set up exactly what you say. We should incorporate lifestyle changes. We should incorporate genomics. We should incorporate new drug treatments. And I want to point out, Dr. Xu has just submitted a grant to enable the better treatment or prevention of diabetes in African American women who are at very high risk. And that includes number one in the intervention. Do you want to say a few things about that?
You're the leader here, but I think we're very happy to have someone like Dr. Sadee at this university who has all this experience in pharmacogenetics, genomics, and we have some new treatments in the diabetes field, which makes it very exciting. But we want to even go farther up the chain to prevent the disease, because once they get the disease, they get all the complications, and by that time, 50% of their 80 to 90% of their beta cells are lost. So we actually can define pre-diabetes where they still have 50% of their beta cells, so we can try to intervene with some novel therapies to try to prevent further damage and going on. So, of course, you all have been bombarded with the lifestyle changes, and we always try to do that, but I think the current evidence suggests with some pharmacological treatment, specifically targeted at the beta cell, that might improve. So the question is, with these databases is, so for diabetes, there's a lot of data on some genes that are associated, but unfortunately they have very little impact in the long run. So we're hoping, though, that with the pharmacogenomics approach, we can maybe identify a better genes that might help us predict who would be responsive to these drugs, and then we can work on a reasonable prevention strategy.
I would respond too to the comments. Perhaps I should have explained that I really had tried to take two approaches. One is make certain that you all understand what's happening out there at the federal level, at the United States level, and also internationally with this whole concept. Here at OSU, in terms of organizing precision medicine, I think we can organize better. And part of, in fact, through exactly these types of events, we need to talk with each other. We have fabulous resources, great scientists. My own sense, having been here just over 3 years now, is that we can actually organize better. We can't take on everything at once, and I think the focus should be genetics, genomics, precision medicine, probably defined through the lens of sequence, using that as a central theme, but not to ignore the critical impact of environment and social impact and other kinds of factors that will ultimately play into this. At the national level, at this Precision Medicine Initiative, they're trying to take on the whole thing. And hopefully this is not a 5-year R01 funding. Hopefully this will be a 10 or 20-year experience. That's what it's going to take to really begin to I think, reap the benefits of this new era.
And I would just finish the comment by that our proposal also involves very heavy public health work, social, economic status, education status, a lot of things trying to predict who will respond to these drugs, not just the genes, but as Dr. Hershberger said, the environment is very important and sort of the opportunities these people have or don't have.
Other comments, questions? I would say that just in follow-up, one of the people in my group will present a study that we have recently funded where it's really not discovery, it's really precision medicine for cardiomyopathy. But there is, the third aim actually is a return of results and actually a very social science type of aim. I think it's critically important too. Should we move on?
Yeah.
Yeah.